So you already see what I will be talking about, combining EPR data, mainly distance distribution restraints, with NMR data to make protein ensemble models, actually. I will use, during the talk, I will use more than one example, but the example that I will use most of the time, you already see here on the title page, because it's a reasonable balance between simplicity and complexity. And this is an RNA-binding protein consisting of two RM-binding domains with a copy-paste error. This should be RNA recognition motif 1, the red one. And the blue one is indeed RNA recognition motif 2. There's a flexible linker in between. And I will show, or I will discuss how one can elucidate what happens to the structure when a short piece of RNA binds. So the binding motif to RM1 is CAU, the binding motif to RM2 is GGA, and here you see the RNA piece, and I will explain how we can make such structures and what are the pitfalls. And before I do this, I want to explain why we want to do this at all, and why we need to combine EPR and NMR data for doing things like that. Let me start from the very beginning. Structural biology for a long time was assuming that there's a single structure of a folded protein, and you can solve the structure of a protein. And this goes back first to crystal structure determinations. People found that some of the proteins crystallize, and then they assumed all of them would crystallize. And this would mean that a single conformer should have by far the lowest energy. This was formulated by Anfinsen, and I call this the narrow thermodynamic hypothesis. He called it the thermodynamic hypothesis. Then the aim of structural biology is completely clear. You determine the structure, and then you're finished with this protein. And about at the turn of the millennium, about 2000, it was found that some proteins do not conform to this narrow thermodynamic hypothesis. Some proteins are or appear to be almost completely disordered, which corresponds to the random coil limit of polymer science. And if you think about this in terms of energy hypersurface, then this Anfinsen domain means you have a single minimum which is much lower by several multiples of the thermal energy than the rest of the energy hypersurface. Whereas in the random coil limit, the energy hypersurface is so flat that the ruggedness is on the order of only the thermal energy, and then everything is populated somehow. Now, since the turn of the millennium, it became clear that it is even more complex. Sometimes you have this case, for instance, for enzymes, that you have thermally excitable states, which are only by, let's say, a factor of 2 or 3 kVt higher than the lowest minimum, and that they are important for function. And what I want to talk about mostly today is that you even can have such rugged hypersurfaces where you have several minima which are all separated by energies which are only a few times the thermal energy. This is the most complex case. And in this case, you really have to describe the system by an ensemble. So a single structure will not do it. And then the question is, why was this hidden for such a long time? And the answer basically is, if you have only a single structure determination method, you will not be able to get the realistic ensemble of such a protein. Now, here's a small, very small zoo of such structures obtained based on EPR, distance distribution restraints, and other data. And they populate the whole continuum of order and disorder. You can have very disordered ones, which are close to the random coil case. You have ones which have only 2 states, like here. And you can have the things in between, more or less ordered. And especially you can have the case, as on my title page already, where a system is rather disordered in the free state, and then it binds a binding partner and can become more ordered without becoming completely ordered. And it is not the accident that all these proteins are RNA binding proteins. I mean, the same phenomena also exist for other proteins. But for RNA binding proteins, they are ubiquitous because you cannot have a single protein for each RNA that you have to process. You have to process very many different RNAs by the same protein, which means that the protein must be a promiscuous binder, must be able to bind different RNA with the same protein. And if you think about designing a system like this, you have to design it as a flexible system. Only then it will work, because the different RNAs have different shapes. And this means if you bind different RNAs, in our case to the SRSF1, the slightly longer one and the slightly shorter one, then you will end up with different ensembles. And it is clear, if to understand how these proteins process RNA, you need to be able to get some structural models. And for this, we need the technique. Now, the first thing I want to show is why we can do this with distance distributions, why we need the EPR part, basically. And here is a picture of one of these proteins. I go back a little bit. This one, HNRNPA1, in this case, not in complex with RNA, just a protein, the free state. And it has one part which is structured, which is again a combination of 2 RNA recognition motifs, and one part which is largely unstructured if the protein is in isolation. And now, how can we find out that this part is unstructured, and how can we find out how strongly it is unstructured? One possibility is to put spin labels on the protein and measure a distance distribution. And here you see what happens in a ordered part, in a folded part of a protein, if you put a spin label. You already have some spatial distribution of the spin label, because the spin label is a side chain, and the side chain also has a conformational distribution. And this means the spin label is not at a single point in space. This may be interesting even if you do NMR, because you have the same problem if you do paramagnetic relaxation enhancement PRE measurements. Then you have a picture like this. The spin label is not at one point. But if you have a disordered region or domain of the protein, the spin label is much more broadly distributed in space. And if you measure the distance distribution between the green label and the orange label, then you get a rather broad distance distribution. And I call this one only moderately broad because this disordered region could be even more disordered than here. Actually, this is already a little bit compact, a little bit sticking to the ordered part. And we could see this because for even between this broad distance distribution and broader distance distributions, we could still distinguish this experimentally. Now, how is this related, these distance distributions, how is this related to the thermodynamic picture? And this is related because each distance distribution restraint basically is a potential of mean force between 2 points in your polymer, in your protein. And you can convert the distance distribution, which you here see with uncertainty estimate, you can convert it to this potential of mean force. This is a distance-dependent energy. This is the distance distribution. You take the logarithm of the distance distribution, multiply with the thermal energy or minus the thermal energy, and add some minimum potential. And then you find that each distance distribution restraint gives you a projection of the energy hypersurface. And this is basically how it works, that you can characterize such a rugged energy hypersurface. If you have enough of these distance distributions, then the projections will give you enough information on this rugged energy hypersurface to make a model. Now, how can we get to these distance distributions? This I have to explain as the first thing. And our main workhorse is this pulse sequence, a rather simple pulse sequence by NMR standards. Basically, if you do solid state NMR, or if you know solid state NMR, this is a slightly modified spin echo double resonance experiment. What you are doing, you have one observer spin, which is the blue spin. You make an echo experiment, and to get zero dead time, you refocus the echo once more. Then you see a refocused echo. And you have a pi pulse on the second spin, which you shift in time. So all the blue pulses, all the observer pulses are not shifted in time. That's a constant time experiment, so that we don't have any contribution from relaxation. But we shift the second pulse. And if I do a pi pulse on the second spin, I flip the magnetic moment of the second spin. This will invert the local dipolar field at the A spin. And this means the resonance frequency of the A spin will just change by the dipole-dipole coupling. And if I shift this in time, then I see a oscillation with the dipole-dipole coupling on the amplitude of the observer echo. Now, from this oscillation, which comes from the distribution of distances, I want to have the distribution of distances. How do I do this? And this is basically a picture made by Luis Fabregas Ibanez, who made the newest software for this. The best way of doing this is separable nonlinear least squares. You have the distribution, you also have the background. So you have two spin labels in one protein, but you have the next protein molecule somewhere in the solution, the next to the next, the next to the next, and they make a background contribution, which is, to first approximation, exponential decay. So you have to fit this exponential decay, you have to fit the distance distribution, and you have a problem that the fitting of the distance distribution is so-called ill-post problem. Very small deviations in the input data, very small noise, can lead to a very large errors in the distance distribution. You will see this on the next slide. So what we have to do, we have to stabilize the problem a little bit. This stabilization is called regularization. So we get a regularization parameter that I will also explain on the next slide. Now we have to fit the background function, the regularization parameter, and we have to perform the regularization to make a fit of the experimental data to the of the simulation to the experimental data. And in earlier times we made this in a two-step approach, and the better way of doing it is the separable nonlinear least squares. You have an outer loop which is nonlinear, where you fit the background and the regularization parameter, and you have an inner loop which is very fast, which is the linear sub-problem, which only makes the root mean square deviation fit of the distance distribution to the data. And this is a constrained linear least squares problem because the distance distribution must be non-negative. It's a probability density. Probability can never be negative. This also stabilizes the problem. This you also have to use. Okay, and here you see what regularization means. So if you would just fit just minimize the root mean square deviation, then the distance distributions that you would get out would be something like this red thing of very narrow peaks. The gray distance distribution is the ground truth, is what we put in this simulated data. And this is what you would get if you just fit the root mean square deviation without any regularization. This is called undersmoothing. And then there's some optimal case. You see, you don't have a perfect fit. You never get a perfect fit for an ill-post problem. but you are pretty nicely or pretty close to the ground truth, which is this gray thing. And then if you increase the regularization parameter even more, then you get to oversmoothing, then you lose features of the distribution. If you have a very large regularization parameter, you completely lose the features of the distribution. So there must be some optimum, and there are different ways of finding the optimal regularization parameter, alpha. And one is based on an information criterion, a Kaigi information criterion. And where this is minimum, you expect you have the best solution that you can get. So basically, the take-home message from this slide is you have to use good software to do this, and you will still have some deviation from the ground truth because the problem is ill-posed. And you have to take this into account when you later model a protein ensemble. I should also mention that there's an alternative way of doing this. If you always work with the same pulse sequence, you can also train a neural network. And this is realized in the DEERnet software by Ilya Kuprov. And what I will now show you on one example for this SRSF1 protein is that it may be useful to use both approaches and compare the results so that you can be reasonably sure that you have the correct result. So I will stop this share and share something else. So this is not the most modern software, but it implements DEERLab and it implements DEERnet. And now I load the data set. And this data set, which I have here, is basically the wild type of the protein. The wild type of this SRSF1 protein, I will explain the protein at a later point, has two cysteines, one at position 16, residue 16, one at position 148. And they are labeled with methane thiosulfonate, and the short RNA is already bound. So here on the top left panel, you see the original data measured by Laura Esteban-Hofer. And this has a real part, it's a quadrature detection, a real part and an imaginary part. Let's switch off the imaginary part. This is how the original data look like. There's a little bit of an artifact here in the end from overlap of pulse excitation bandwidths. And here's the first estimate of the distance distribution from a very simple model. Now, we use regularization and the neural network approach. I just do it automatically. And this is how you should do it unless you are an expert. You just do it automatically. And again, I have to make a stop share and share a different window so that you see it. Now let's see. I have to find this. Okay. Now, hopefully you see this window and you already see the experimental data fit and the background fit. And this has been provided by DEERnet, by the neural network approach. And to get an estimate of the error of the uncertainty, 32 networks were separately trained with different training sets. And the variation between these 32 neural networks is used for getting an estimate of uncertainty. And you already see here that the fit is rather nice. Now, at the moment, the program is working on the regularization. The regularization, because it's a nonlinear least squares, is a little bit slower than the DEERnet. But in some cases, it can be useful to have both of them. Here you see the original Tironov with a first estimate of the regularization parameter. And here the green one that you see is the regularization parameter, which gives the best agreement with the neural network. And then you already see you get two slightly different estimates. One from the regularization, the green one, and one from the neural network, the blue one. And at the moment, the program is computing the uncertainty for the Tikhonov regularization. And this will still take a little bit of time. Let me see. Yeah, we are at, you cannot see this, we are at 72 out of 96. So what I will then do, or what the program will then do, it will make a consensus of these two distance distributions and their uncertainties. And then one will see how well the two agree and what is the best estimate. Now again, I have to make stop share and share a different window. Let's see this one. And then you get a report like this, PDF report. And this shows the two distributions with their respective error estimates. And you see they don't completely overlap in their respective error estimates because there's also some model bias. And then this is the consensus distribution with the uncertainty estimate, which still tells you something. This distribution for this particular double mutant or for this particular spin-labeled mutant is not very broad. And we will later use this in modeling of the ensemble. Now, at this point, let me get back to the talk. At this point, we have to think about something else, namely the spin label. And this is a problem that you also have to solve when you work with paramagnetic relaxation enhancement. So these spin labels that you use have rotatable bonds in the side chain. So they have a dihedral angle distribution. And the one that is most used, this methane thiosulfonate label, here attached already to a cysteine, has 5 rotatable bonds. And now if you make a molecular dynamic simulation, then you see that the distribution of these dihedral angles is semi-discrete. So for each dihedral angle, you have a certain number of canonical values, and then it's distributed narrowly or a little bit more broadly around these values. And the model that we use for accounting for this in protein modeling is called the rotamer library. So we say we combine this angle with this angle, with this angle, with this angle, with this angle. This is a rotamer. And then we try to find as many rotamers as we need to get a good model of the label. And here you see an artist's expression of this. The green balls are the midpoints of the NO bonds, of the nitroxide bonds, where the electron spin is. In space for the different rotamers, and the volume of these green spheres is the population of the rotamers, which you can find out when you model it like this. And what you also see here is how precise or how inaccurate the approach is. This is basically a test on T4 lysozyme, how well we can predict where the spin label is. And this is like a GPS localization of the spin label. We had measured, or others had measured 5 distances from other spin label sites to this site. This site is 131. And then from a GPS-like algorithm, you can find out, based on these 5 distances, where you would expect the spin label to be. And this is the simulation. And basically, you get the accuracy of about 3 angstrom. Aha. Now, maybe I also want to show how this works. So again, I stop share. I go to one of our modeling programs. This one has a graphical user interface. And here you already see an ensemble of SRSF1 with the RNA, the bound state that I showed you on the title page. And now we want to make the spin labeling and compare the distance distribution to the one which we just made with the other program. And for this, I select the residues. Chain A is the protein. Then I take all models of the chain and the residues that we had spin labeled in this protein, for which I showed you the distance distribution. I first select this. And then I compute this RNA-binding library. And then this starts to run. It will only take half a minute. So now for all protein conformers in the ensemble, it computes the rotamers for both labeling positions. And after this is done, which is now, we have a complete distribution of the spin label for all of the conformers. Then we have to attach the spin label, which I now do. And now at this point, we can compare the result for our protein model with the experiment. Again, I have to change screen share. This is another window of the same program for DEER simulation. So we have one label at residue 16 and another label at residue 148. Oh, Porphyry effect. So I checked this before, but for some reason at the moment this doesn't work. Okay, good question. Why not? So then I have to go back. Sorry, I did check this yesterday, but I cannot show this. So in principle, you would now have seen that this fits the data, but I have this on a later slide as a plot. Okay. Now, once you have these distance distributions and once you can model, you want to make a model of the protein. And first I show you a general pipeline, which would be used with whatever modeling program you use. First, you need to analyze your experimental data, the distance distribution that I showed you, your NMR data. All this has to be analyzed and has to be translated into restraints. Then, if you want to make an ensemble, you have to make different conformations of the protein. This would be conformer generation. After this, you may have 10,000 or 1,000 or 20,000 conformers, and you have to find out which ones are the best ones. And this step is called ensemble reweighting. And here you use all your restraints. At this point, you can use only distribution restraints, but here you can use all the restraints, for instance, also NMR restraints, PREs, maybe RDCs, and so on. Then after this, you have a representative ensemble. You may have different data to validate what you have, because you will always get a result. The result may be wrong. You want to establish that it is a sensible result. So you have to compare to additional data, or you have to make additional experiments. And then at some point you deposit it. And here is the program MMMX that I use for the ensemble models that I showed today. This uses models for the rigid part of the protein that can come from cryo-EM or X-ray crystallography or NMR, or nowadays predicted by a neural network by AlphaFold, or by machine learning by AlphaFold. Then first you make some preparation, you cut out parts of the model, and then you compute rigid body arrangement, you compute the flexible parts, you make this ensemble reweighting, which is called ensemble fit in this program. And at the end, you may also want to make some ensemble analysis, which helps you in the validation. And now I will talk you through this kind of modeling pipeline. And for this, the first thing I have to introduce is the regiflex approach. So the problem is, if you have this rugged energy hypersurface and these ensemble models, you have a complicated problem. Let's say you have a protein with 380 residues or so. Looking at all possible conformations is impossible. If you think about the conformation of a peptide chain, then you have different Ramachandran angles, and each of them can have different realizations. And let's say you have 370 Ramachandran angles, or pairs of Ramachandran angles, each of them has 3 possible values. Then you get a number of conformers that you could never generate in a computer. So you have to have some way to simplify the problem to get the representative result. And one possibility of simplifying this problem is to say, let me go back. Parts of the problem of the protein will follow this Anfinsen dogma. Parts of the protein will be folded, and we will consider them as rigid bodies. And only parts will be flexible. You can also see this here. Parts are like a rigid body. And you can establish that they are by making NMR measurements, for instance. So what I need to have are 2 things. I need the relative arrangement of the rigid bodies, and I need conformations of the flexible linkers. And this is simplified because the flexible linkers are shorter, which the number of conformers that you should consider is exponential in the number of residues. So you have a much simpler problem if you have short flexible linkers than when you consider the whole thing as flexible. So first we solve the rigid body arrangement, and it turns out that we can do this by having 3 reference points in each rigid body, because then for each pair of rigid bodies you can measure 9 distances, but there are only 6 degrees of freedom, 3 translations, 3 rotations between 2 rigid bodies. So the problem is overdetermined if you have 3 reference points. So first we solve the rigid body arrangement, then we make the flexible protein peptide linkers, and then if we have RNA, we make the flexible RNA linkers. And I will talk you through this by an example. Now, the first thing that we have to solve is this rigid body arrangement. And we want to be sure that we have seen all solutions that can exist. So we need something called exhaustive sampling. And because the problem is large, because we have so many different arrangements, we have to find some way of doing this. And this again uses distance distributions. Let's say the black line is the experimental distance distribution. The red line is a Gaussian fit. Then I put a lower bound at minus full width at half maximum of the Gaussian, and an upper bound is plus full width at half maximum of the Gaussian. And in between, I say all the possible distances are. So I neglect something like this, which may be also an artifact of the distance distribution. So in between, I put my sampling points. Let's say I put 3 sampling points. I have a digital resolution. And then I combine all possible sampling points. Then I have an exhaustive set of, let's say, a few million of rigid body arrangements that I should consider. Of course, then I only have a certain distance resolution and a certain resolution in this sampling space. So I need to have some refinement to get to the correct distance. That's one point. The second point is I have a few million of these possible combinations. How can I solve the problem? This works by distance matrix geometry. Let me go back. We have only a few reference points. Let's say for 3 rigid bodies, we have 9 reference points. So we have a problem that we can solve, a very small geometric problem that we can solve with distance geometry, which is simple matrix computations. This can be done for a few million sampling points in a relatively short time. So this is how we solve this problem. Then we have to create the flexible linkers. And for this, we start from the statistics that we have for the dihedral angles in the peptide chain, which is the Ramachandran plot. So we have residue-specific Ramachandran plots. And if you have additional information at this point, then you can use, for instance, alpha helix or beta strand propensities from NMR to tweak the statistics. But this statistics at the moment is not used for fitting everything, only for improving sampling statistics. And here again, the distance distributions are very useful. This is again a Gaussian model of a distance distribution. And now I make a conformation and I look at the distance between two residues that I have already computed. And if the distance is inside the distance distribution, this is a good conformer. If it is here, it is still a reasonable conformer. If it is here, that's a poor conformer. So for each distance distribution that I have, I can compute the probability that the conformer is consistent with this distance distribution. I multiply all these probabilities, and then at some point I will find out, oh, this conformer is of so low probability that I can discard it. And I can do this before I have completely generated the conformer, which enhances the sampling, which makes the sampling of conformation space more efficient if I have distance distribution restraints. A similar thing can be done for RNA. For RNA, you don't have Ramachandran plots, but an American group in 2012 made the pseudo torsion angle model of RNA. And then you can do basically the same thing as for the peptide. When we did this, and this is for this SRSF1, you can sometimes see something like this. The RNA threads through the protein. This is another protein where we computed with Sienna, a modeling program from NMR. And there you can see the opposite thing. In this case, the protein, the flexible part of the protein threads through the RNA. Both of this is unrealistic. So this means you also need some analysis in your modeling to recognize such cases and to discard these conformers. Okay. Now at this point, I have something like 10,000 or 2,000 conformations, and now I have to find the best possible ensemble with, let's say, 20 or 25 or 100 conformers. How do I do that? So at this point, I can use all the restraints, also the NMR restraints, for instance, PREs. The NMR restraints you can only predict if you have the whole ensemble. So the prediction from a single conformer doesn't help you because you measure ensemble restraints and they are on average, they are just a single number, and you cannot establish whether a single conformer is consistent with these restraints, but you can establish whether a whole ensemble is consistent with these restraints. But the question then arises, how do we combine restraints from different techniques? This is not easy. For instance, if you have PREs, you probably want to look at the root mean square deviation between the experimental PREs and the back-calculated PREs. If you have a distance distribution which stems from the solution of an ill-posed problem, then root mean square deviation may not be a very good figure of merit. What you rather do, what is rather robust, is to look at the overlap between the simulated distance distribution for your whole ensemble, the red line, and the experimental distance distribution, the black one. And the overlap is the area that they have in common. And if both distributions are normalized to a unit area, then this overlap, which is 79%, 0.79 in this case, gives you an idea how well your model, your ensemble model, agrees with the distance distribution that you have measured. Now here we have an overlap criterion. For the PREs, we have an RMSD criterion. For a Sachs curve, we have a chi-square, which is very similar to an RMSD, but not exactly an RMSD. And now we have to combine all this. And on top of this, we have the problem that the experiments were done under slightly different conditions. So this was measured on a frozen solution. The NMR was probably measured at 25 degrees Celsius, or because the measurement was long, you had to cool down to 4 degrees centigrade. The SAX was measured as a different concentration than the NMR. So the restraint sets between different techniques might be inconsistent. And you also have to account for this. How do we do that? So we balance the subsets of a heterogeneous restraint set in the following way. First, we fit each individual restraint set. We fit only the distance distribution, let's say, with the overlap criterion. We maximize the overlap, minimize the overlap deficiency. We fit all small angle X-ray scattering data by minimizing chi-square. We fit all PRE data by minimizing root mean square deviation. And then we do this individually. So for each restraint set, we get an ensemble and the best fit that we can get for this restraint set. And then we combine all restraint sets and we make a balanced fit by minimizing the so-called loss of merit. So each restraint set has its own figure of merit, overlap for the distance distribution, root mean square deviation for the PREs, chi-square for the small angle scattering data. And this figure of merit increases when I combine the restraint sets because they are slightly inconsistent. And now I have to have an ensemble which fits all of them. And this means this ratio between the balanced fit and the fit of a single subset can never be smaller than 1, only larger than 1. I subtract 1. I sum this over all restraint subsets. I divide by the number of restraint subsets, and I call this loss of merit. That's a number which is always larger than 0. If it is larger than 1, then probably you have very inconsistent restraints. If this is something like 0.2 or so, then your restraint sets are consistent. Here's an example, in this case, not an NMR example, in this case, distance distribution and small angle X-ray scattering for this HNRNPA1 protein. And here you see you can get quite decent overlap of the distance distributions at the same time, good fit for the SAX data. A little bit worse than you would expect. Chi-square of 2.016 is not very good, but this is what you can do for systems like that. And the remaining time I will use for an example on the protein that I already showed on the title page, this serine arginine rich protein. And first I want to explain why we looked at this protein. So this protein is involved in several aspects of gene expression. So it binds to the spliceosome. So messenger RNA, mRNA has to be spliced. So there are sometimes even alternative splicings for the same mRNA to make different products. And you need to have proteins that are doing this. And this is the spliceosome. And SRSF1 binds mRNA and brings it to the spliceosome. It's also involved in microRNA processing. It is involved in mRNA decay. If the mRNA should not survive for too long, it should decay after some time. You also need proteins that do this. It is involved in export of mRNA from the nucleus to the cytoplasm, because translation of the mRNA to proteins happens on the ribosome. The ribosome is not in the nucleus, it's in the cytoplasm. And you can also have mRNA decay in the cytoplasm. And because this is an RNA-binding protein, which can interact with several other partners, it is involved in all these different things. So that's why it's a very interesting protein. Here's the basic structure. You have two different RNAs, RNA recognition motifs, which are rather promiscuous. So they can bind, they recognize certain binding motifs, but many different RNAs have these binding motifs that can bind to these RNAs. Here's our construct. We actually cut off one of the disordered domains. We have a flexible linker here. We have the two folded parts, which we treat as rigid bodies. We remove two tyrosines for reasons of making it more stable. And to keep it in solution, we attach the GB1 and the histec via histec. Okay. Then, first thing, before we do any modeling, we should get an idea whether we see something reasonable. And here you see the protein in the absence of RNA and the protein bound to the short RNA, which I introduced in the beginning. So it has one binding motif for RM1, one binding motif for RM2, and it's rather short. Here are the reference points for this Rigi part. So 3 labeling positions on each protein. So these tyrosines we can also use, we have mutated them anyway, we can also use them as labeling positions. And this is a native cysteine, which we mutate out if we don't want to label here, and otherwise we use it. This is the second native cysteine. This is the pair which I showed you before. And these are 2 residues that we mutate in RM2. Now, here you see distance distribution that we measure for the free form and for the RNA bound form. You might not have expected this. Sometimes the RNA bound form has a larger distance, also it's rather short, but it may orient the 2 rigid parts so that you get a larger distance. Sometimes, this is the one that I showed you before, the distance distribution that I made. Sometimes the mean distance doesn't change very much, but the distance distribution gets narrower by binding of the RNA. Sometimes it shifts and gets a little bit narrower. Sometimes it shifts and it remains broad. That it remains broad means that you don't get a complete disorder to order transition upon binding of the RNA. Okay, now I want to explain how we applied this Rigi flex approach to this protein. First, free form. In the free form, we have 2 rigid bodies. So we measure these distances between the reference points and we make a rigid body arrangement. And then for each arrangement of the 2 rigid bodies, we create a model of this flexible linker. This leads us to a large number of possible conformations of this protein, let's say 1,000, 2,000, and then we make the ensemble fit and reduce this. So here is the script, how this looks like. We have this Rigi module in MMMX. Actually, you can find under mmmx.info, you can find the manual of this program. Here we have the Rigi module. We add the rigid body model that we already made with the prepare module. We separate the rigid bodies. We say, okay, we want to have 50,000 trials, which is enough for 2 rigid bodies. After these 50,000 trials, we want to have 2,000 models. We define our reference points and what kind of spin label we use. And then we provide all the distance distribution restraints that we measured. Between pairs of levels, we have more than this. We have all the ones that we need. And we also say already, we have a linker between the 2 rigid bodies, between residue 87 and 121. These are 34 residues. Because this already restrains the rigid body arrangement, the linker cannot be longer than the number of residues times 3.8 angstrom. That's a completely extended beta structure. The linker can never be longer than this, and this already helps to solve the problem. And then we say we superimpose everything on rigid body 1. This would be the whole module, the whole source code that you need, the whole script code that you need to solve the rigid body problem. Then we have to create this flexible linker. And also in the flexible linker, we have a label position 107. And this means we have a few distance distributions between a reference point in one of the domains and the linker. Here I give only Gaussian restraints. I have an N-terminal anchor of the linker, a C-terminal anchor of the linker. I have the sequence of the linker. I say which residue numbers these are. And that's the whole script code that you need for flex. This expand means we expand the rigid body arrangement that we have. So we do this for all rigid body arrangements that we have. For all the 2000 that we generated in this step, this is done. And then in the end, you have to make the ensemble fit. Here we add all the models that we generated in the flex step. This is a technical detail. And we say where we want to save the ensemble, and we give again all the restraints. And now here we have the NMR restraints. These are PRE restraints for the same spin label at the singly labeled protein, singly labeled at the native cysteine 16. This is the NMR frequency, and there are a few more parameters. The timing of the init sequence that you have in this experiment where you measure the PRE, and some estimates on the mobility and on the relaxation times. And then you can fit all this. And here's where you would find this in the manual, in the online manual. Okay, here's the result. So the black lines and the gray bands are the experimental distance distributions, and the red lines are the fits for the ensemble. They are a little bit more rugged because the ensemble is not very large. They are very broad distributions. And you want to look at the ensemble in the end. You don't want to have too many conformers. But by and large, except for this very narrow feature, which we cannot sample, the agreement is quite good. Now, here's the PRE restraint fulfillment. It's not perfect. You sometimes see that the PREs are a little bit off, but this is what we can get in this case from the back calculation. Now, here is the ensemble made in this way for the free SRSF1, and here's the ensemble made in this way for the bound. This is what I already showed you on the title page. And here you see that everything is superimposed on RM2. You see that the arrangement of RM1 to RM2 is already broadly distributed. And you also see the linkers all over the place. The cyan thing is the linker all over the place. If you bind the short RNA, of course, this has to become more ordered. But again, if you superimpose everything on RM2, you still have disorder in the relative arrangement of RM1, and you also still have disorder of the linker, not as much disorder as you had before, but still disorder. Okay. Before I wrap up, I have one more case, because I want to show you can also do this with the programs that you may already know from NMR work, not only with MMMX. And here I show a pipeline that is combined from Sienna, which you might usually use in NMR. And in the last step, only for the ensemble fitting, we use MMMX. And in between, we use a program Yazara to refine the Sienna structures. The Sienna structures usually don't have extremely good, the raw Sienna structures don't have extremely good protein geometry, clashes, Ramachandran plot, and so on. And Yazara is used to help with this. This is another protein which is involved in the replication of some viruses. And these viruses use a backdoor to translation in the human cell. Called internal ribosome entry site, and this is a piece of virus, which is just this internal ribosome entry site for the encephalomyocarditis virus, EMCV. And we wanted to have the structure of this in iris, which helps the virus to replicate its RNA and to express its proteins in the human cell. Okay, here we have 3 rigid bodies. Again, RNA recognition motifs 1, 2, and 3 and 4 from 1 rigid pair. This can be established by NMR. And we have some secondary structure of the RNA piece in this case, and we know from other experiments where the stem loops of this RNA bind to the protein. And we use all this information. We have the in Sienna to make 20,000 models in Sienna. We select the 10% that have the lowest target function in Sienna, so we still have 2,000 models at this point. And here you encounter the problem that I showed you before. You can have threading, that the peptide linker goes through a stem loop of the RNA, which is unrealistic. And it also turns out this may also happen in Sienna if the problem is complicated, that sometimes you get unrealistic bond lengths in the RNA sugars. And so we throw out a few of these models. Then we have initial Sienna models. I think this were 567 or something like that. We refine these one by Sienna. Aha, here it was still more than 1,000. We make again the test whether we have threading or inconsistent bond lengths. And then we make the ensemble refinement, and the ensemble refinement we make with the MMX. In the end, we have 25 conformers. How well does this fit? Now, in this case, we also have small angle scattering data. Here you see the fit. It's not as good as it was for the SRSF1 for the distance distributions. And here you see the problem that I showed you before, that you can have inconsistency if I fit only the distance distribution. For this particular pair, I have an overlap of 83.7%. If I fit it together with the small angle scattering restraints, the overlap drops to 68.4%. Because one is measured at low temperature, one at room temperature. In one case, I have labels, I have different concentrations, so it may be slightly inconsistent. So I get a consensus model from doing this. Okay. At this point, I've talked for an hour. I just want to acknowledge people. You have already seen the names on the top of the slides. Maxim Yulikov was involved in the supervision of many of these projects. Daniel Klose knows a lot about protein modeling, was also involved. The work on HNR and PA1 was done by Irina Reach. The work on PTBP1 and EMCV iris was done by Christoph Gmeiner. And the work on SRSF1 was done by Laura Esteban Hofer. And the DEER lab for getting these distance distributions, this was developed by Luis Fabregas Ibanez. And we have a long-standing collaboration with the group of Frédéric Alain in biology in ETH. And especially for PTB1, the work was together with Georg Dorn and Tebbe Fran Vries, and for the small angle scattering, also Emil Dedic. SRSF1, Christina Nguyen was involved, and Antoine Clery. And I'm afraid I don't have, here, for HNR and PA1, Elisabeth Lehmann and Leonidas Emanuelidis were involved. And the funding came from ETH and from the Swiss National Science Foundation, and I thank you for your attention.
Thank you very much, Gunnar, for this really nice talk. Just for the attendees, you can find the question and answer window at the bottom of your Zoom, or just in your Zoom controls. And please type your questions there so I can see it and read them out. And to give you some time, I guess, to ask them, I can maybe start. So when you showed the DEER lab and neural net, you get some kind of error estimate in the distributions, and they are not necessarily the same. And can you comment on how much does this influence the actual calculation in the end? Is this even that important, or?
To some extent, this is still an open question. So for the protein ensemble models, we do not yet have a complete uncertainty estimate. I can tell you why the problem is complicated. So we have the distribution of the conformation, and then on top we have an uncertainty of the distribution of the conformation. And basically, to solve this problem, we should have not only one ensemble model, we should make a large number of ensemble models and then see how much the different ensemble models deviate from each other. And we are not yet at this point. We are working on this. And in doing this, of course, to make the different ensemble models, we now would need to consider the uncertainty of the distance distributions, for instance, also the uncertainty of the PREs. Maybe you see this at one point here. So for the PREs, you see we also have error estimates. And then basically in this balancing, we would have to consider the error estimates. And this also means we should probably make for distance distribution an error estimate which involves several analysis procedures, which maybe also involves an error estimate coming from the models that we put in. So this is really still work in progress. And I would expect that it will take a decade or so before we can really have good error estimates for ensemble structures.
Okay, then we have a question by Tamara Wolf from the chat, but I just read it. So perhaps this is a very basic question, but does the regularization method in DEER affect the distance distribution significantly?
So basically, I can show this on this slide. So basically what you see when you increase the regularization, you broaden the distribution. So this means whenever you have a regularization, the distribution will be a little bit too broad. You also see this here. So you can never get if you have noise or some other deviations, you can never get the perfect, the ground truth back. You will always have a little bit of broadening. And now, depending on what criterion you use for the regularization parameter, you will get a slightly different determination of the optimum regularization parameter, and this means you will get slightly different broadening here. This is one effect that you have. Another effect is if you do regularization, then basically all these peaks will have about the same width because you apply the same smoothing. You also see this here. The narrow one is more strongly broadened than the one which is already broad. And this is one point where the DEER-net is better. The DEER-net is not really regularization. The DEER-net, in such a case, can find a narrow peak and then a broad distribution. But there are other problems that you have with the DEER-net. That's why we always recommend to try both of them.
And then there's another question in the chat. Will the matching between SAX and DEER improve if they will be measured in the same temperature?
Once more,
if the matching between SAX and DEER improve if they are measured at the same temperature?
Well, unfortunately, we cannot measure the DEER at room temperature. And I mean, there are some papers who say they measure at room temperature. Then we cannot go to such long distances because the relaxation is too short. And measuring at room temperature would still mean that you have to immobilize the protein, which would again make the experiment different from the SAX. So we have to immobilize the protein because we measure dipole-dipole interaction. And if the protein moves fast, then the dipole-dipole interaction is averaged out. Vice versa, the SAX measurements cannot be done in frozen solution or not as easily in frozen solution. So if you could do a low temperature SAX and could get good data, then maybe this would improve. On the other hand, what we have checked in one case, in the HNRP-A1 case, is that the freezing, and also in two cases actually, also in fused in sarcoma, that the freezing doesn't seem to change the conformation very strongly, but this may be different from protein to protein.
Okay, then we have one question from Dinar Abdullin. Thank you for the exciting talk. How do you take into account the errors of the spin label positions in the final modeling? At which modules of MMX is this done?
So basically, again, I have to say we don't have a complete error estimate at the moment. We say these errors of the spin label positions, they will contribute to the uncertainty of our ensemble model. And what we basically rely on at the moment, I mean, a few minutes ago I said, okay, we don't have the uncertainties of the models. What we know for these broad ensembles that we have, the 3 angstrom is much smaller than the distribution of the conformations that we have. So if I shift the ensemble model, each distance in the ensemble model by 3 angstrom or 6 angstrom, then I wouldn't see much difference in the ensemble model. So in a way, we live from the fact that the proteins are disordered. If the disorder is much larger than our uncertainty, then we can make a reasonable model. This is also one of the reasons why we don't use these distance distributions so much to make structures of ordered proteins. Even if you cannot crystallize an ordered protein, even if it's too large for NMR, structure that you could make by EPR distance restraints would be worse quality than a high resolution structure that you could make, for instance, by cryo-EM.
Okay. The audience seems to be a little bit shy today, but I have still one more that may make sense. So work-wise, what do you say is time limiting at the moment? Is it still like the biochemistry, or is it the EPR
I think the wet lab is limiting. So there are strange dreams to say, okay, maybe we could have robots that make the protein expression and the spin labeling. But from my experience, this is not realistic in the next future. Each protein is an individual. You have to, it's not so much the manual work of making additional mutants. You have to optimize the protocols. You have to be careful to check that everything is okay. This is not so easy to automate. So in the end, you need someone who makes the mutants, checks the mutants, makes the protein, makes the spin labeling, and this takes time. So this restricts you to maybe in one doctoral thesis, 50 restraints or something like that. And this means in one, if you have proteins of this size, then you may need something between 20 and 50 restraints, and then you can make maybe 2 structures per thesis, but not much more.
Okay. Thank you for the interesting talk. Did you try alternative machine learning methods to neural, like the principal component analysis?
No, we didn't. Actually, I have to say that the machine learning method, basically this was work by Ilya Kuprov from, who just raised his hand. Maybe if Ilya raised his hand, then maybe Ilya should answer that.
We should be allowed to talk.
Thanks, Gunnar. In fact, the PCA methods, so essentially singular value decomposition is another way to stabilize these Hadamard unstable problems. The people who tried that a lot is Jack Fried's group in Cornell. What they have discovered is that it has a lot of fiddle factors, which are just a different instance of, the fiddle factor. That's the regularization parameter. You have to decide at which singular value you truncate, and that's a very unobvious manual decision. Yes, it has been tried. Madhur Srivastava and Jack Fried tried that. Yes, it's reasonably successful. And just as every other method, it does require you to be a bit of a specialist in what one is doing. And so it's really hard for, an inexperienced person to pick these parameters.